Great. I want to thank the organizers for inviting me to talk today. This is a big topic, and we do large-scale analyses of many thousands, hundreds of thousands of enzyme sequences and structures in order to get some traction on this problem. One of the main ways that nature evolves enzymes for new functions is that it reuses what we call a set of privileged structural scaffolds. And those are structural scaffolds where the ancestral type has been used over and over again to evolve many different reactions. And what's conserved in these proteins is not what they bind, it's some fundamental aspect of the chemistry that they do. So here are just a couple of examples showing that multiple folds, we see multiple folds that can do this, and it's a big set of proteins. We estimate over a third of the enzyme universe is represented by these functionally diverse enzyme superfamilies. Being able to look at this at a very large scale gives us some new kinds of ways to ask questions we really couldn't ask before. And that includes some of the questions here where we want to know how nature's actually remodeled these structures to enable new chemistry. What are the changes that have to happen? What's the range of reactions these proteins can do? That's a very useful way to think about the problem if you want to learn from nature how to perhaps get new information for enzyme engineering. Applications include predicting molecular functions of enzymes that are found in all the different sequencing projects. It's a very hard problem in many ways, and we have very little experimental information about these. So here's an example of one of those superfamilies, one of the best characterized ones that serves, I've been working on for many years, as a matter of fact. The enolase superfamily has about 50,000 sequences. We know roughly 23 different reactions that are as different as the reactions you see on the screen. And those are quite different reactions. These proteins that do these reactions can be well less than 10 or 15% identical, the sequences. And they range across the biosphere from archaebacteria to higher eukaryotes. What you see in the structure superposition here is superposition of the conserved active site residues in the crystal structures of these 6 proteins. And what you notice is it's an enormously similar active site given the diversity of these proteins. We now understand that what this active site evolved to do was to abstract a proton from a carbon, alpha to a carboxylate, leading to a similar type of stabilized enolate anion intermediate. We find that little substructure in the natural ligand of every one of the proteins we've looked at, either from sequence or structure or function. And what that does for us is it constrains the search space for functional inference for all 50,000 sequences we have. Fortunately, we're able to pick up that motif because it's highly conserved across even the most diverse of the 50,000 sequences we have in this set. So now we can identify new sequences, sequences from metagenomics projects. If they can be attributed to this superfamily, we know something fundamental about how they work. This sort of approach has, for us, provided important clues for predicting functions, especially for what we call outlier enzymes. And those are enzymes, frequently dehalogenases, enzymes that do biodegradation types of reactions, hard chemistry for a biological system to do. But when we can assign them to one of the superfamilies, and each of these represent a different superfamily, we get we have gotten enough clues from our models of the superfamily that along with other biological information and experimental information, we've been able to predict those functions. Predicting it's all well and good to know what these have in common, and that's very useful. It's much more difficult to predict the specificity determinants that make them different. And here you see the same set of superimposed active site residues, and I've shown now in the liganded crystal structures the ligands that are found for each of these 6 enzyme types. a different color for each enzyme, and that little substructure that they all have in common, the alpha carbon from which the proton's abstracted, is very locked in its pose in that active site. The rest of the molecule is all over the place. So our job is to figure out what the specificity determinants are that determine binding as well as subsequent reaction steps in the reaction of each of those proteins. This turns out to be partly difficult in part because we're looking at enormously divergent proteins, and we see different rates of evolution of the sequence families for each of these different reactions. And what that means is we can't use similarity as a proxy for functional boundaries because they evolve at different rates. That greatly complicates that problem. Some of these proteins also have superfamilies are promiscuous in their reactions, another complicating issue. And the last one, I think, is a little bit of a subtle point, but important to recognize. Each superfamily is unique and different and has different ways of evolving new functions. And that means we can't identify a general level of identity at which annotation transfer is safe, except at very trivial levels where you're very, very similar sequences. Even there we see some exceptions. But as we start to get more distant, where the really interesting stuff is happening, and where we want to know what's the differences in those sequences and structures that specify different reactions, this gets very difficult. For those of you who haven't done these kinds of computational studies, I want to just give you a general strategy. We start out by identifying highly divergent homologs. There are great tools and data resources for this. We don't have to spend our time on that anymore. We then cluster the families into subgroups or smaller families that we hope might represent reaction families, monofunctional families, using a variety of tools. And then we'd like to be able to map conserved elements of sequence to topologically close area in the active site, and then map to that any functional or mechanistic information we have that helps us try to deduce what the differences in these motifs might be about. And that's sort of the general way we do it. Now, the way the field is moved now, of course, is to add many orthogonal approaches to try to get more useful data for getting those clues about function. And I've listed a few of them here. Ultimately, at the end of the day, you have to do an experiment to find out if you were right. As you'll see later, the number of experiments we can do is vanishingly small compared to the size of the data. So we have to have good ways to pick dark targets to characterize. So the rest of the talk, I'm going to discuss some technology that we've been working on to help us with these global studies, some of the opportunities that gives us when it helps us to ask that we haven't been able to before, and challenges at that very large scale level. And then I'm going to give you one example of some work we've been doing to find functional boundaries in one small superfamily. So most of the superfamilies we work on are way too large to work on using traditional methods. Some of them will have more than 150,000 sequences. That's simply, in terms of data management, it's a significant issue. Practical limits to really look at and learn something from a multiple sequence alignment, you're talking maybe 100 sequences, not too many more than that. High quality phylogenetic trees, there are tools now to do very large phylogenetic inference, but it still depends on a very high quality multiple alignment, typically requiring structures to get the alignment as good as you can. That's also very challenging. And as I mentioned before, members of these superfamilies can be enormously diverse so that you have to have sufficient structures to get a reasonable alignment. We've been using protein similarity networks. I don't have time to spend much time on it, but if you think an analogy to a tree where you have 3 different colors representing 3 different functions, you can also represent this by graphical network models. we use thresholded models. So each circle or node represents a sequence or a structure or a group of sequences that are redundance filtered. We draw a line or an edge between nodes if the similarity is as good as some statistical significance cutoff. And just as, so we can dial the stringency of the threshold up or down to remove edges or add them back. And that gives us a very nice interactive way viewing these networks by Cytoscape to explore that space. Okay, so here's an example of one of those networks for the radical SAM superfamily. I've simply colored these by our subgrouping sets. They aren't functions, they're just the best similarity boundaries we can find. We could color these by many, many different kinds of information and start to ask which of these groups sees each other the closest, which are the furthest away, how big are they relative to each other, and then we can drill down for much more sophisticated questions. Here is the same network viewed at a more stringent similarity cutoff, and now you take the network apart. What you hope is that each color represents a group that might have similar functions. This is not, getting the sweet spot for that is also difficult. I don't have time to talk much about this today, but we're hoping to move toward more automated methods for doing that. Okay, one of the first observations we make for these superfamilies is how little we know. This is a network of all the glutathione transferases, key in secondary metabolism, metabolism of xenobiotics, et cetera. Everything in gray we don't know what they do. Everything with a color has been assigned a classification that is based on similarity. Of 13,000 sequences represented here, we have 92 physiological functions known, and this is one of the best studied superfamilies that exist. The GSTs also have a problem that there has enormous reaction promiscuity. They evolved to handle a lot of different ligands, so they can do reactions on a lot of different ligands, and that means that many of them will be able to do many reactions. The difficulty in trying to figure out the physiological functions, you don't know what substrate to test with. So there's a set of, I think, about 15 or 16 now that you can test with and try to assign them to these classes, but it doesn't tell you anything about what they actually do. We see many kinds of structural variations that are associated with specificity determinants. Here is a picture of topologies for the alkyd phosphatase superfamily. There's a core Rossmann fold domain, many inserts. Those inserts are not homologous to each other in many cases. We know they track with some general functional features, not specificity, but we don't know how they do that. So that's kind of the state of the field at the moment. Many superfamilies have enormously complex domain structures. Here we see for the radical SAM superfamily a domain structure network. This is not based on similarity. It's just there's a green vertical bar at the center that's the SAM binding domain, and these are all the different variations in domain structure. Again, if we're trying to sort these into subgroupings that make sense, we have to take this into account, and it's a big issue. What we've learned about 20 of these superfamilies that we've been able to study in some detail is that the large-scale analyses really does add a very powerful context for interpreting and predicting the range of reactions the superfamily can support. Mostly, we find out that we knew very little and that there are probably a lot more reactions there than we had thought about. Prediction of reaction specificity is very difficult. And especially this point I want to emphasize again, different superfamilies evolved in really different and unique ways, using different kinds of variation in structure, active site, domain organization of the proteins, and you have to have some idea of what that's about to be able to even start working with these data. What we do is we collaborate with experimental labs with deep knowledge in some of the proteins in our superfamilies, and we help them to find targets to go after that will get us into some of those new questions. Now, the tautomerase superfamily, this is a small superfamily. It has only about 15,000 sequences. This is a collaboration with Chris Whitman at UT Austin. There are 5 known reaction families. These are interesting because they're very small. The monomeric, the smallest form that acts as an enzyme, the sequences are 60 to 80 amino acids. They all apparently use an N-terminal proline that's required for activity and key to the mechanism. We've found some exceptions to that actually in our study that I'm not going to have time to talk about today, but it's a really interesting wrinkle on the story. As I mentioned, some of the outlier reactions that we get clues on by learning something about these superfamilies, this cis-CAD protein is involved in degrading insecticides in soil. There's a bacterial system that's a little pathway that degrades that insecticide, rendering it inert. We, in part, were able to interpret the mechanism, or Chris was. We didn't do that. We did the computational part, knowing that this protein is part of this superfamily. Turns out that sequence similarity relationships, in fact, track with the known named reaction classes we've identified. That doesn't mean all the proteins in each of these groups, colored groups, do that reaction. They're just namesakes. But we do know some of the very different reactions that are represented by those namesakes. We have been interested for some time, in part because of the uniqueness of the cis-CAD reaction, which is shown in green here, what the specificity determinants were that distinguish the cis-CAD from the tautomerase reaction. And I've identified 2 proteins with an asterisk on the network for which we have mechanistic information and structures. So how do you start working with these kinds of proteins? Well, the first thing you may want to do is align the structures. These proteins are only 18% identical, so aligning those sequences is not terribly useful. If we do a structure-based alignment, we learn a couple of key points. And the first one is that the orange guys, which represent the tautomerases, are only half the size of everything else in this superfamily. And it's the C-terminal half of that duple version of the proteins that are represented in all those other subgroups that the tautomerase aligns with. So that's very useful information. We also know that there's a highly conserved arginine that's required for activity. A better way to do this is to use all the information we get from the network. So what we did is we computed the shortest similarity path between the tautomerase enzyme, characterized enzyme, and the cis-CAD, and then did a structure-based alignment of those. And that's what you see on the right side of this slide. So you basically see the same thing I showed you in the last slide, but now you've got sort of enzymes that have what we think are really look like transitional features in those contemporary enzymes that give us some better clues about how the differences, what the differences between the cis-CAD and the tautomerases really mean. Oops. So the first hop that we see is going from this tautomerase enzyme to the fused tautomerase. It turns out that there are some duple-sized tautomerases in this orange group, which wasn't known before. And what we see is, we see the fusion. We see there's a little insert here that is associated with that fusion. So far, that fused tautomerase is actually quite similar. It's 40-some% identical to the tautomerase that is anchoring this. The second hop, we label number 2. That when we start to see some changes, we see the loss of this insert, which we don't see in any of the other sequences that we've computed between these two. The next hop is especially interesting because you can see by the red arrows an addition of most of the machinery needed for the cis-CAD reaction. Now, there are certainly other changes in the sequences that I can't go through in detail here because of time, but those are the key spots that we know are positions that we know and residues that we know are critical for cis-CAD activity. We then go to the fourth hop, which gets us to linker number 1 up here, which is the closest to the cis-CAD. And we see here the introduction of the tyrosine machinery that is critical for finishing, that is also required in the cis-CADs. We don't see it in the tautomerases. We have some preliminary kinetic analysis from the Whitman lab where we see full activity for the tautomerase, less tautomerase activity for the fused guy. So it's probably doing something else with a different substrate. We see less yet with linker 2, and we see 3 orders of magnitude loss of activity with linker 1, but we can still get a little bit of tautomerase activity. And when we get to the CGX or the cis-CAD, both of these are very similar to each other. We don't know what the CGX does. We've lost tautomerase activity entirely. We haven't done the reverse experiment yet because of some difficulties with actually doing the assay, and we're not sure that, well, we think the cis-CAD, we know what the cis-CAD substrate is. We think there are many different substrate specificities here. And so unless we can get a better idea of what those specificities are, which is truly difficult, we're going to have it'll take a while to get those experiments. But we're very enthusiastic about this kind of approach to give us an idea of what sort of a speciation view of variations between different functions, so we can key in on the critical variations that we see. So the next steps, of course, are to build this out and study it in more detail. So the first thing we want to do is expand this linker set. We have 2 ways to do that. The network that you see is what we call a representative network. So we've taken all the sequences that are identical at least 50% or more and put them in one node because we had to compress the network to be able to visualize it and work with it. So each one of these nodes, including the hops, may have several hundred different sequences. We can add those back in just in this little region that I've shown by the black oval and study that in much more detail. The second thing we want to do is phylogenetic reconstruction because it's been thought for a long time that cis-CAD, the subgroup, evolved from the tautomerases. There's some sort of speculative information that supports that. We only see the short guy and the tautomerases. The only RKL sequences, and there are quite a lot of them in this superfamily, are found in the orange group, the tautomerases. So we'd like to get a better handle on what the tree looks like and see what clues that may give us. That's given the diversity of these proteins, building a tree is really a hypothesis, even a really good tree, but it still gives us some clues for how to think next. We also want to add in metagenomic sequences to expand that set. We haven't done that yet. Basically what we do is we build a hidden Markov model to the set that we're interested in, and then we go fishing in metagenomic data here from JGI. But I can give you an example for a different superfamily. This is the nitroreductase superfamily, and any of you who heard Eyal Akiva's talk on Monday, he talked about this superfamily. So what Eyal did is he took one small subgroup from his superfamily that had about 270 sequences out of a total of around 15,000 sequences. And he took the hidden Markov model that describes the profile that describes this small subgroup and ran it against the IMG/M set. And we go from 270 sequences here to over 10,000 here. Now these have been filtered for length and a number of other characteristics that would make them look weird or not very good matches to that HMM for one reason or another. But you can see these are the sequences that come from public databases. You can see where they map on the larger network that includes the metagenomic data, protein sequences so far. We see brand new subgroupings that likely have quite different capabilities. And we're really excited about this direction. The size is staggering. If we try to go from 15,000 sequences in the whole superfamily and add that metagenomic data, that's going to be a network that is way too big to study, for a user to study on their own computers. But we can take our subgroups and do that, and they're manageable. So we're very pleased about this direction and looking forward to doing these kinds of things in the future. So this is just one example of one way to drill down into this kind of large-scale data to get new clues about sequence structure-function relationships for many different applications. So I'm going to stop here, and thank you for listening, and I'm happy to answer questions.
Okay, thank you. That was great. We have time for maybe 2 or so questions.
Great talk. So I had a question. So you're talking about getting more, if we can get to the point with the similarity networks that you can automatically define the threshold, can you get to a point where you can then use that structure, superfamily structure, to add some kind of statistical relevance to propagation of annotations? So you can say, based on experimentally characterized proteins and then given our network structures, then we can say with a certain level of confidence that yes, it's okay to propagate that annotation, no, do not propagate that annotation.
So the answer is no, and that's not because we can't automate the processes. It's because we really don't know, we don't have enough information about any one of these superfamilies to know how well the knowns can be transferred, that annotation can be transferred. This is a big question for, I don't know if you have heard of the CAFA contest where a lot of people are trying to predict function of proteins. This is the kind of challenge that we have. Now, where we have a fair number of queries that we know the function, and we've done this with the GSTs, with Richard Armstrong's lab, he cloned 87 GSTs that were in that unknown space, got structures with Steve Albo for 27 of them, and simply tested them for one of the promiscuous reaction, disulfide bond reductase, and found they were all over the superfamily. So now we at least know that we can't assume it's just in one small group. So we can get answers like that. Sometimes we can do some of that sort of transfer, but we typically have to have quite a few characterized proteins to bracket what we think might be that functional boundary. I think we're ultimately going in that direction, but it's a very hard problem.
Hi. Just wanted to know relationship between superfamily and EC numbers. So, for example, is first three EC numbers are enough for superfamily, or superfamily may have different? So, how many numbers are enough to define?
It's a very good question. It could be all 4. So I wrote a current opinion 10 years ago, worrying about using EC number to transfer annotation. And in these superfamilies, it's a big problem. Now, that said, typically people are looking at much more similar proteins than we are. But generally, across a superfamily, you can have all 4 digits of the EC number represented. And certainly, you will have many different digits at the 3rd position, which defines the overall chemistry. Well, if you have something close enough, you can make that transfer, but you don't know how close that is. You might for a particular superfamily. I'd be willing to make guesses for particular superfamilies we've studied, but It's hard otherwise.
Okay, I think we have to move on to the next speaker, but I think Patsy will be around
Okay.
during the break for additional questions.